Okay, so I'm a research scientist in the Max Born Institute für Nichtlinearoptik und Kurzzeitspektroskopie. This is part of the Leibniz Society, so the German institute in Berlin. And my research topic predominantly is about proton transfer processes. So these underlie basically the basic processes of acid-base chemistry. So because we want to measure electronic structure or transient electronic structure, and we want to do X-ray spectroscopy to learn about that, and doing experiments at this machine here from SCS is actually a superb way to get get insight into and get successful measurements also to do this kind of experiments. The main important thing is that you do these experiments at ultra-fast timescales, so femtosecond, few picosecond timescales, because these are basically the timescales where you have the elementary steps in proton transfer, and we want to be really at those timescales to really get the most out of these processes which we want to study. Because we do it in water, because this is the medium of life now, but it is first of all general context is that proton transfer is being used by life. Now you have signaling and you have proton gradients in living matter, so biological cells, and the transmembrane proteins control that. And the proton transfer is basically at the heart of these biological machines. The other thing is that proton transfer is also used in the technology of hydrogen fuel cells, so energy research basically. And if you want to also study the full of it, then you also want to study the microscopic processes. And this is our next step to learn more from and get more details out of these processes. And maybe we will also be helpful for also that people want to get more use of this in applications. The huge machine here is only part of it now, because you have a couple of kilometers accelerators behind this now. So ultimately this is necessary to prepare the X-rays. This is at nitrogen or oxygen K-edge, because these are the atoms and the elements in the chemical compounds which are part of these acids and bases. And we want to follow this by doing this X-ray spectroscopy, and this is then happening here. You have in the target chamber a liquid flat jet, which basically is used you excite it with a laser to initiate a proton transfer process, and then you follow it with X-ray pulses and see in course of time what kind of changes are induced. Now, the tool we use is that we use photoacids. These are weak acids in the electronic ground state, but when you can excite them with UV light, then they become suddenly very strong acids, and then you initiate a proton transfer process which you can then follow in real time. And this is at the heart of the principle of our experimental procedure. Yes, actually it did work really well, I must say. We are really happy. And then our enthusiasm and happiness actually also resonated with the people who are actually responsible for this whole machine. Because then, they also put a lot of effort in it to get this stuff going. And when our success is also due to their effort and their success in getting these things done at European XFEL.